Hello, and welcome to everyone joining us for this X Talks webinar. Today's talk is entitled ADCs in Oncology Trials Practical Strategies for Clinical Development Success. My name is Corey Stanton, and I'll be your X Talks host for this event. Today's presentation will run for approximately 60 minutes, and the webinar includes an interactive Q&A session at the end. To make the most of this session, feel free to submit questions and comments for our speakers throughout the presentation, and we'll try to attend to as many as possible. If you're joining us via GoToWebinar, you can use the questions chat box, which is located in the control panel on the right-hand side of your screen. And if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. And if you're viewing this presentation from our LinkedIn Live event, you can submit your questions using the Comments tab on LinkedIn. Please note that all audience members are in listen-only mode and that this event will be recorded and made available for streaming to those who registered on xtalks.com.
At this point, I'd like to thank Worldwide Clinical Trials Oncology
To develop the content for this presentation.
Worldwide Clinical Trials is a full-service
Specialty Contract Research Organization, or CRO, built to serve the global biotech industry. Backed by leading retention rates and a culture rooted in its core values, Worldwide provides customers with teams experienced across all functions, knowledgeable in complex drug classes and study designs, and with data-centric methodologies that help bring next-generation therapies to cancer patients. You can connect with Worldwide on LinkedIn. And now, let's take a moment to meet our featured speakers for today's discussion. Guiding our panel today is Andrew Zupnik. Andrew has dedicated over 25 years to oncology, leading study optimization, delivery oversight, training, and innovation at Worldwide Clinical Trials Oncology. A cell and molecular biologist with a PhD from Columbia and a BS from MIT, he began his career at Prolog Research before guiding Novella Clinical's oncology division to market leadership. Following Novella's acquisition, he spent nearly 7 years advancing oncology-focused solutions at IQVIA Biotech. And also joining us today is Dr. Udayan Guha. Dr. Guha is a physician scientist with over 15 years experience in clinical development across academia, pharma, and biotech, specializing in targeted therapy, immunotherapy, and cellular therapy. Before next year, he was VP, head of clinical development at TCR2 Therapeutics, and earlier led first-in-human immunotherapy studies at Bristol Myers Squibb. At the NCI-NIH, he directed a bench-to-bedside lung cancer program, advancing research on tumor heterogeneity and resistance to EGFR TKIs. We're also very excited to welcome Stephanie Kordahu. Stephanie is a senior clinical operations professional with experience in biopharma, CROs, and academia, specializing in oncology and rare diseases. She has led phase 1 through 3 trials globally and regionally across ADCs, monoclonal antibodies, and cell and gene therapies. And next year, she built and leads clinical operations, driving excellence, risk management, and patient-centric focus, while strengthening site relationships and optimizing trial execution. Stephanie holds degrees in neuroscience and drug development. And rounding out our panel today is Dr. Shodinde Coker. Dr. Coker is Executive Medical Director at Biohaven Pharmaceuticals with expertise in translational oncology and early phase clinical trials. Previously, he advanced antibody drug conjugate programs at Bristol Myers Squibb. He earned his medical degree from the University of Ibadan with postgraduate training in clinical pharmacology at the University of Aberdeen, internal medicine residency at Howard University, and hematology, oncology, and clinical pharmacology fellowships at Dartmouth. He is board certified in internal medicine, medical oncology, hematology, and clinical pharmacology. Welcome to all our panelists, and before we get underway, we do have 2 interactive poll questions for our audience members to respond to in real time. Appearing on your screen now, we'd just like to get a sense from our audience members who are joining us, which side of industry you're joining us from. And your options are academic site or government, sponsor, biotech, sponsor, large pharma, investment, or simply other. And again, we're just looking to get a sense from our audience members who are here with us today for this presentation, which side of the industry you're joining us from. Are you from an academic site or government? Are you sponsor biotech, sponsor large pharma, investment, or other? And you can click on any of those options and hit the submit button. Thank you to our audience members who have already voted. We'll leave the poll open for a few moments longer to allow us to get a majority vote from our audience here today, and then we'll go ahead and take a quick look at those results to see who's with us for our presentation. Excellent. Thank you so much to our audience members who have voted. I'll leave the poll open for about 10 more seconds. So if you haven't had a chance to vote, make sure you click on any of those options and lock it in with the submit button. Fantastic. Thank you, everyone. Let's close the poll now and take a quick look at those results. So, in response to our audience members here today, we have 46%, our majority are in the sponsored biotech space. But then we have 43%, so nearly half our audience in the other category, and 7% of our audience academic site or government. So, thank you to our audience members who are joined us here today. We have one more poll question for you before we launch into our panel discussion. This next question for you is just to get a sense of your level of familiarity with the ADC space. And our options here to assess your familiarity is, are you seasoned? For instance, you've brought ADCs to market. Do you have growing familiarity? Are you actively developing ADCs? Are you interested? So not currently working in ADCs. Or finally, very bewildered in ADC, what's that? And you're just here to learn. And again, we're just looking to get a sense to your level of familiarity with the ADC space. Are you someone who's seasoned in the space? For instance, you've brought ADCs to market. Do you have growing familiarity? Are you actively developing ADCs? Are you interested, not currently working in the ADCs? Or finally, you're not sure how you ended up here, but you're looking into ADCs and you're excited to learn. Thank you to our audience members who have already voted. We'll leave a poll open for about 10, 15 more seconds to get a majority vote from our audience. So if you haven't had a chance to vote, make sure you click on any of those options and hit the submit button to lock it in. Fantastic. Thank you so much, everyone. We'll leave a poll open for about 5 more seconds. So if you haven't had a chance to vote, make sure you click on any of those options and lock it in with the submit button. Again, we're just looking to get a sense of your level of familiarity with the ADC space. Fantastic. Thank you, everyone, for voting. I'm going to go ahead and close the poll now, and let's take a quick look at those results. So in terms of our audience's familiarity, we have 61%. Our majority here today have a growing familiarity, so they're actively developing ADCs. But then next to that, we've got 21% who are interested, not currently working in ADCs. 11% of our audience here to learn about ADCs, which is great. This should be a great discussion for you. And finally, 7%, a small minority here, are seasoned in the ADC space and have actually brought ADCs to market. So with that, I'd like to hand the mic over to Andrew to kick off our discussion. Have a great presentation, everyone.
Great. Thank you, Corey. And thank you for everyone who submitted some feedback on those questions. It helps us understand kind of the audience and customize some of our conversation today. We're really hoping to make this interactive conversation across the panel. And thank you to everyone for joining wherever you are in the world. We have folks that I can see from all kinds of time zones. So really excited to join you and to have this really esteemed panel of medical and operational experts join us to kick us off. And I appreciate folks who were honest on some of those questions about familiarity with the ADC space. I want to make sure we level set on what we're talking about. There's a lot of acronyms in our industry, right? So ADCs in the form of antibody drug conjugates. Let's just level set on some terminology and some background, and then we'll kick off with some conversation around where the industry has been moving. So as a scientist, right, ADCs are at its baseline level, been around for a long time, since the 1980s. With antibody engineering. However, in the past few years, we've seen quite a revolution with ADCs, specifically in oncology, new linker technologies, new payloads, and you see ADCs regularly in the spotlight at industry events with some really groundbreaking and practice-changing results. A couple of data points for our audience. There are now 14 FDA-approved ADCs on the market today. But this has been a very hot space. For those who are currently working in the area, you know what I'm talking about. Those who are here to learn, hopefully this gives you some data points. There are over 1,100 ADC therapies in development, quite a lot. There are nearly 3,000 clinical trials underway globally as of last month. And then we've seen a very significant spike in investment. R&D investment has more than doubled in the past 5 years. It's about in the $10 to $12 billion range annually. So we're excited to talk to you today about this really hot area. During the webinar, we'll explore the evolving landscape of ADC development. There have been a lot of advancements just the past couple of years. Even this past weekend at a conference at World Lung, ADCs were in the spotlight and some recent data in lung cancer. We'll talk about scientific, clinical, and regulatory design challenges, for those who are looking to get into the space, things that to think about as you move into the clinic, and then some considerations from operationalizing and running your trials as we think all the way to the registrational and approval space at the end of the development spectrum. So I'm really excited to join you today with a panel of seasoned clinical and medical experts who will be sharing their experiences. This is really an interactive kind of best practices and lessons learned conversation. So let's jump right in. I think, and I'll try to bounce this around and make sure everyone's jumping into different questions. We've got a series of questions lined up to walk through as the panel, and you can think about it as the continuum of overall drug development. So let's start with some terminology clarification. ADCs are a pretty unique drug class. They're different from targeted therapies, they're different from immunotherapies, and there's a lot of ADC specific terms that we use. So Udayan, maybe we can start with you. Can you help with some of the definitions? When you're talking ADCs, you hear terms like target and linker and payload, or even DAR and therapeutic window, bystander effect. I mean, these are novel concepts for folks who've not worked with this drug class before. Can you give us a bit of a rundown on some of those terms?
Yeah, thank you. I can start off. First of all, thank you, Xtalks and Worldwide, for providing me this opportunity to participate in this webinar. So I'll start off with some terminology. So ADC is essentially targeted chemotherapy. It was initially portrayed as a magic bullet. The term started by Paul Ehrlich in the early 1900s in the context of antibiotics and microbes. Uh, and uh, and it is very relevant to ADCs, obviously, although that concept has been challenged since then. And you will see during this talk why I say that. Now, ADC consists of obviously a monoclonal antibody that binds the target. Now, the target expression has to be high in tumor cells and low or minimal or ideally no expression in normal cells. So these are essentially tumor-associated antigens or tumor-specific antigens. Now, this antibody is carrying some payload that is essentially the chemotherapeutic agent that has high potency in killing tumor cells. But for that matter, then it's also highly potent in killing normal cells. So that's where the antibody goes and binds the target and takes these payloads along with it. And the chemical moiety that links the payload to the antibody is essentially the linker. Now, there are a lot of novel linkers, and so a lot of development in ADCs in the linker side and the obviously also the payload side. Now, DAR stands for drug to antibody ratio. Essentially, it's the number of molecules of the payload to one molecule of antibody. Like, for example, the most common are like a DAR of 4 or 8. Sometimes it is 2. So now, just one more word about the linker. We'll come back later also, I'm sure. But the linker should be stable in serum because ADCs are injected intravenously, right? So once the ADC binds to the target on tumor cells, they are internalized. And then there has to be a mechanism of releasing this payload inside the tumor cells. And that is done by various mechanisms based on the platform. And one key ones are the enzymes that are there in late endosomes or lysosomes. What enzymes cleave the linker to release the payload differs in different antibody platforms, I mean the ADC platforms. Like, for example, cathepsin B that cleaves the VC linkers, like the peptide linkers, glycosidases, glucuronidases, and others. Now, there are a few approved ADCs with non-cleavable linkers also, and in those cases, the whole ADC molecule must be metabolized or cleaved, processed to finally release the payload.
Thank you. Perfect. How about your view, Dende? What about some of the terms like bystander effect or therapeutic window? I know that was new to me moving into the ADC space.
Thank you, Andrew, and again, thanks for having me here. So by the therapeutic window, we're talking about the concentration of the drug that effectively kills the tumor cells and spare normal tissue. So the whole concept of ADCs is to actually widen those windows, that window. The highly potent cytotoxic drug that are not ADC platforms really have a narrow therapeutic window, and the whole aim of ADCs is to widen that, concentrate to the delivery of the cytotoxic agent into the tumor itself. As my colleague Dr. Ryan said, this is a very evolving and very exciting space in drug development. With regards to the bystander effect that we see, we're talking about the payload being taken up by tumor cells that express the antigen. Once the tumor cell is lysed, the cytotoxic payload is able to kill surrounding tumor cells that do not express the tumor associated antigen, and that's the bystander effect. This has its downsides as well, because once the drug diffuses into the tumor microenvironment, it's actually able to kill not only the tumor cells that do not express the antigen, but also normal cells as well.
Beautiful, thank you. And I think that's where we're seeing a lot of innovation in this space, right? There's a lot of different variables you can play with, the antibody design, the linker, the payload, et cetera, and maximizing all of those nuances. So maybe I'll start with you, Dende. A follow-up question there. You know, some of the drugs that are already on the market versus things that we're seeing now in development with, like I said earlier, thousands of drugs, ADCs in development, thousands of active clinical trials. There's only a handful of variables you can play with. What are you seeing as some of the newer trends? Where has the industry been moving versus what's already been on the market so far?
Yeah, thank you. As my colleague said, ADCs, they are not a totally novel concept. The earlier payloads were dominated by the microtubule disrupting payloads such as MMAE and DM1. While effective, these agents often carried very significant toxicities, and that limited their clinical utility. More recently, we're seeing a shift towards topoisomerase 1 inhibitor payloads, such as SM38 and deruxtecan. And these have anchored several ADC approvals. Today, the majority of ADCs in development are really gravitating towards that payload class. With innovations in payload chemistry and playing around with the drug antibody ratio, the DAR ratio, they're designed to create better efficacy, bystander effects, and really optimize the delivery of the payload to the tumor. At the same time, we're seeing emergence of next generation formats. They include biparatropic ADCs. Essentially, you have the ADC that's able to bind to different epitopes of the same tumor antigen. We also have bispecific ADCs, where you have two different antigens being targeted by the same ADC, and you have dual payloads, one antibody and two different payloads attached to it. And this is all designed to overcome resistance of the tumor and overcome tumor heterogeneity as well. So really an exciting time to be in drug development in ADCs.
Agreed. You, Diane, you've seen a number of different payloads in the development over time. What's your take on the payload side of things?
Yeah, no. So, I mean, that was pretty extensive answer by Dr. Coker. I just add that the first ADC approved was in the year 2000, right? So now you can see like how much development has happened, and it was for a CD33 targeted ADC, gemtuzumab ozogamicin, also called Mylotarg. Now that had a different history, like it was taken off the market and again brought back. And again, all this is because of the earlier payloads and the ADCs, the toxicity and all that, right? And the specificity is getting better and better. And that has something to do with that. Then obviously a lot of MMAE ADCs were approved. Now MMAE is great potent toxin, but also there are a lot of long-term side effects like peripheral neuropathy and all that, right? But as Dr. Coker mentioned, the topoisomerase inhibitors are the most popular now. Now that targets a lot of tumor types. Essentially, it's a topoisomerase 1 inhibitor, and it prevents the resealing during the DNA repair. So a lot of strand breaks happen in the DNA, and that leads to cell cycle arrest and finally cell death. The good thing about the topoisomerase is like the toxicity wise, you can go to higher dose levels. So at the end, it's all the payload that is killing the cell. So if you can increase the concentration of the payload, so that's part of it. And that's the reason why a lot of ADCs are now getting approved for topoisomerase.
Agreed. Certainly seeing that on my end, on the service provider side. We still see MMAE based trials. There's a place for that, and then there's a heavy focus in the Topo 1 payload and even next generation versions. And like we were talking about, we're seeing new twists on linkers and targets and other variables that you can do with your antibody engineering. I mean, Diane, what are you seeing as far as targets? I think we've seen there's been a couple quite well validated ones like HER2. Now we're seeing a ton of different
ADCs that
Yeah,
are in
so
development?
that's a very important point in this ADC, right? Obviously, the first thing is the target, right? Where is the monoclonal antibody going and binding? And there are not many validated targets, as we know. The ones validated for obviously solid tumors are HER2, TROP2, and then there's Nectin-4 now, and DLL3 is coming out for small cell. But you can count the number of targets. For hematological malignancies, CD33, CD30. So where are more novel targets coming up? And yeah, I mean, a lot of biotech is working on, but then again, like maybe we are all gravitating towards more validated targets because there's a lot of risk in a new target. So the question is like, this is something the whole community have to think that we need new targets for that discovery needs to happen. And another thing about the target is not just high expression in tumor and low in normal. That's the first thing, right? But also that target has to be internalized well, and that's where all these validated targets come in. And so there's a lot of good research need to happen to get additional targets.
Agreed. How about the linker side of things, Dende? I mean, that's the other variable that you can play with, and how tightly it holds onto your payload effectively is there's a lot of different options there.
Yes, precisely. And like my colleague said, the linkers are generally classified into cleavable and non-cleavable linkers. The cleavable linkers, really exploits the tumor's own physiology, acidic pH or proteolytic enzymes to release the payload. Examples include acid-sensitive linkers or protease-sensitive linkers. We're seeing a shift towards non-cleavable linkers. These linkers, such as thioether linkers, they're highly stable in plasma, and advances in technology have made this really possible. These linkers, they rely on lysosomal degradation of the linker and the tumor microenvironment, and the tumor itself to release the payload once it's internalized. So the advances in technology, more stable linkers, is where I see the industry shifting to. And for the most part, most of the ADCs, they're working in, tweaking that linker technology to optimize the chemistry of the ADC.
Beautiful. Thank you. I can talk about antibody design all day being a scientist. Appreciate you guys' input there. For those on the call who are here to talk about studies and operations, maybe we can shift into that conversation. I welcome everybody to come to the World ADC Conference, which is in a couple of months, if you want to have an entire week talking about manufacturing antibody design, as well as clinical trials. So let's now move into assuming that you have a new ADC. And you want to bring it into the clinic, right? I hear a lot of questions around, obviously there's plenty of ADCs and historical knowledge to build off of, but with all these variables, payload, linker, target, DAR, how do you think about selecting a starting dose and moving into your first in human trial? I hear a lot of questions on that front because we obviously don't want to be in dose selection and dose optimization for 2 years. How do you focus that out of the gate? Maybe you, Diane, we can start with you and then go. From there.
Yeah. So the starting dose selection for ADC is, I would say, relatively straightforward. A GLP tox study has to be done, which is an IND enabling study, and that will determine the highest non-severely toxic dose in the most relevant species based on the target and everything, or two. Mostly it is in cynomolgus monkey. And the human equivalent dose of that HNSTD in cyno is calculated based on allometric scaling and PSA and all that. Essentially, it has to be divided by 3.1 to get the human equivalent dose of HNSTD. Now, based on ICH-S9 guidance, a safety factor of 6 is taken into consideration for the maximum recommended starting dose, MRSD. For example, if the HNSTD is a 30 milligram per kilo, which is very typical of a topo payload ADC in cyno, then 1/6 of HED, like human equivalent dose of HNSTD, will be around 1.6 milligram per kilo of starting dose. So that has been the starting dose of several of the even the approved topo payload ADCs.
Perfect. Thank you. For those who are scribbling notes quickly on that, you'll have the recording afterwards, and you can access some of the specifics. Stephanie, we're talking operations finally. Maybe I can turn it over to you from an operational perspective. How do you think about that very first in human dose escalation starting point?
Yeah, thanks, Andrew. So, dose escalation can look very different with the various ADCs in terms of starting dose and dose levels or ranges that might be tested, especially like take consideration MMAE versus topo, like we were talking about earlier. So, in general, ADCs really tend to have a small threshold between activity and toxicity. This is something that I think we'll hear probably over and over again today. The FDA really is strict on dose escalation because they've seen these toxicities and what can happen with certain ADCs. So the approach to dosing really can be quite different. You may decide to do actual body weight. Some ADCs now are getting more into using adjusted ideal body weight. And a lot of times that can help mitigate some of the risks we're seeing with toxicity or dosing too high based on body weight. Dose frequency is also something that they'll want to consider. That's another factor that we will build in. But really, truly understanding what your preclinical data looks like, as Dr. Guha mentioned, and getting the right starting dose is important. And being able then to explore and open to exploring alternate dosing strategies once you're in the clinic and you actually see that data start to emerge.
Yeah, I completely agree. We have the fun job of seeing quite a number of ADC trials here on the worldwide side, and I'll say many of our first in human protocols are evolving in real time. Like you said, you may explore different dosing regimens, and that's all part of dose optimization as you go, right? You have an idea to start, like you said, and then be open to exploring multiple options as it evolves. You may be in dose optimization and escalation for a little while just to get really clean on that final dose before you move into your specific, kind of phase 2 arms. Yeah, maybe Stephanie. So what about what other things? You mentioned some of the FDA feedback. What are other considerations when you're thinking about study design? Certainly in today's era of Project Optimist designs and dose optimization, and there are a lot, I'd say, quite a number more variables in thinking about a first-in-human trial. And so, what are some of your recommendations from a pre-IND and a regulatory and study design planning perspective?
Yeah, thanks, Andrew. So, usually the FDA is going to lean on the data that they've seen from these prior approved or current ADCs. And they will likely use that to then advise you during pre-IND or IND on the design and dose selection strategies that you might present to them. As the drug developer, it's really important to understand your competitive landscape. As you mentioned, there are thousands of ADCs being developed. So really getting into what those dose schedules look like, what toxicities have been observed, and any proactive measures that might have been taken in those other studies in ADCs with similar payloads or linkers can be important. And the FDA may, and we've experienced this, they may ask that to see data from your lower dose levels before you escalate above a certain dose level in dose escalation. And that's something that will take a lot of preparation. So making sure that you are getting the sites ready for a specific data cutoff, that you have all the PK, ADA, safety, PD, all of that data that you need to be able to support that, hey, our ADC actually is different and it is safe to escalate above a certain dose. But they may also advise you, which can be tremendously helpful, especially if this is your first time developing an ADC, on what actions or modifications to take if you do see certain toxicities that have been observed in similar ADCs. They can advise you on overall safety monitoring of patients. And one thing I would say to look out for specifically with ADCs is you'll probably get some feedback from the clinical pharmacology reviewers. And so they'll definitely have a lot to say about your dose justification, and clinical pharmacology development plan. So those are all things that you'll want to take into consideration and make sure you address appropriately during your pre-IND and IND stage.
Yeah, that's good feedback. We have a couple of trials here where that was exactly the FDA's input of we want to, we want to look at the historical, we know what the historical averages are with those payloads. And so please come back to us when you get to that point. And, they really were uncomfortable. Although there was a novel linker technology, for example, that made sense that you could dose higher levels, they still wanted you to come back to them before you moved into that space. So really good input. Maybe I think there's a good topic for all of our panelists. Maybe Diane would love your thoughts on some of the study design considerations early in design.
Yeah, I mean, that was pretty extensive. Thanks, Stephanie. But I'll just add that, before a GLP toxicity study is done, which FDA will rely on as a IND enabling study, right? You should try to conduct a properly designed dose range finding study. And that is essentially to plan your GLP tox, like what doses you're planning and all that, right? Now, FDA will, again, as I said, rely on the GLP talks, but the properly conducted DRF study is very important. The other thing is the toxicokinetics and the PK from the GLP tox study, and that will decide, obviously, also on the schedule, right? Whether it will be Q3 week, which is very popular, or Q2 week, or even weekly, and all that kind of dosing. Now, another thing that is often you have to keep in mind is the PK in my studies where you are doing the efficacy studies and a proper exposure response relationship in these preclinical models and then project it to humans so that you know at what levels you are expecting efficacy, right? So FDA is going to look both the toxicity that is coming from the GLP tox study and also these minimum efficacious dose that you are projecting and how good you can project it with considering all the problems with mouse modeling and all that we already know. But these studies are very important, and that's why, and some of these you should also show it in the pre-IND, because then if you have a good package, even in the pre-IND, then you get a lot of advice from the FDA. And we always recommend having a synopsis or even a draft protocol. Like more you give it to them, then you get more advice. Otherwise, they are just going to say that, hey, come back to us when you really file the IND because we don't have the protocol or the synopsis. So all these things should be well documented and planned for the first in-human study.
Agreed. How about your perspective, Dende, from early regulatory and study design planning?
Yeah, great points by Stephanie and Diane there. I could just, build on Dr. Aguha's points with two anchor questions. Is your target antigen, is it validated, or is it novel? If it's novel, you want to have a clear understanding of its expression levels in the population, heterogeneity, and then also the payload class. Is the payload class novel, or is it validated as well? How you answer these questions really determine, questions such as the patient population that you'll be studying the drug in, whether you need a biomarker strategy going in, and also the starting dose rationale, whether it could be more conservative or take more risks with that. So those questions really determine, what direction you're going to go. So for example, if you have a novel payload class or a new payload in a well-validated class, you might want to start your starting dose level at a very conservative level, have a more cautious dose escalation. Also, you want to pay very close attention to your PK, your biomarker results as you start early on in the trial.
Yeah, agreed. I think that hits on a question I get a lot here, and having the benefit of seeing, over 10 phase 1 ADC programs, dose optimization and how to, let's move into that space, right? So we've got our design, we're in the clinic. How do you think about dose optimization today with all of these variables you guys were just talking about? Maybe we'll go down the same kind of three of you guys. Stephanie, what's your view on how best to do that? I get that question all the time.
Yeah. It's a good question. I mean, you really want to identify a couple of dose levels for, dose escalation and backfill. And you'll want to really look at the totality of your data: safety, tolerability, preliminary efficacy if you have it, PK, and any PD data that you've collected or analyzed so far as well. A lot of us are now familiar with the Project Optimus, and so that really encourages the tailoring of your dose or dose regimen in specific indications versus the old school thinking of a one size fits all dose and taking that into part two. Overall, preliminary dose exposure, safety, and efficacy relationships are going to be important to review as well as part of selecting doses for dose optimization. And one thing that we found especially helpful has been doing real-time PK modeling and simulation, because this can really help support the selection of dose levels by understanding what your Cmax and your AUC look like at each of those doses.
Yeah, good input. Udayan, what's your take on the optimization side of things?
Yeah, I mean, the Project Optimus is, I think, quite relevant, especially for the ADC studies, because, the therapeutic window is pretty narrow, as we know, right? But even before going into the Project Optimus, like where you can choose 2 dose levels or schedules or, sometimes 3 dose levels, even before that, as in dose escalation, one thing that we have seen time and again in different ADC development is the intermediate dose level that you have to test, right? So you start a phase 1 study with a dose escalation, but then you see that, hey, we are getting this kind of toxicities at this dose level, maybe an intermediate dose level should be tested. Now, those kind of nuance in the dose escalation and uh, uh, testing different intermediate dose levels or schedules is very important even before Project Optimus, which is generally in the part 2 of a phase 1 study. Sometimes there's a phase 1/2 study in the 2 part, or sometimes even phase 2/3 big studies. Like a classic example is the Cadherin-6 ADC from Merck Daiichi. They are testing 3 different dose levels as a phase 2/3 program. And this kind of things at some point, I think in the ADC development, FDA would ask us to do.
Agreed. How about Dende? What's your view?
Yes, dose optimization, as we've all heard, is a very important part of ADC drug development. But my opinion, it's less about statistically powering to see differences, but more about seeing if there's a trend, a directional trend in what you see with regards to differences in safety and efficacy. And we can really bridge some of that gap with, PK/PD modeling, exposure response modeling and biomarker analysis. Randomization is recommended, but I have heard of anecdotally of some sponsors who have been able to argue their way out of randomization in that dose optimization um, phase of drug development, and really relying on adaptive design. Like Dr. Diane said, you start out with 3 doses and then you drop one. So, I think there's still a lot of uncertainty about how to precisely dose optimize, and we're really learning as we go along.
Agreed. Perfect. How about for some of the audience? I saw we had some folks who are well in the clinic or pretty seasoned. How about real quick, Dende, on the later end of the drug development spectrum from a registrational study design perspective, what are some of the hurdles that you've seen or some of the recent trends there?
Yeah, that's a great question. Some ADCs have, they've pursued an accelerated approval registration strategy for some indications, which means that, confirmatory studies are needed. But we have seen, for example, with Mylotarg that Dr. Diane mentioned earlier, these confirmatory studies don't always pan out and would have to be withdrawn.
Yeah, Udayan, I think,
Diane, yeah, I think so.
yeah, you had a good case study that you were talking about the other day.
Yeah. So, I mean, developing an appropriate assay and cutoff for screening target, if that particular target needs prospective screening, is a huge issue in the field, right? Because when you start the phase 1 study, you don't know how the target is working or the target response, expression response is working. So you choose some arbitrary cutoffs based on preclinical studies. And one example I want to highlight is the approval of Elahere, mirvetuximab soravtansine, right? This ADC, it's a folate receptor alpha targeting ADC that took more than 8 years for the first accelerated approval. And initially, a phase 3 was conducted named Forward 1. And in this study, folate receptor alpha expression was greater than 50%, 1 plus, right? So uh, PFS hazard ratio in this study was 0.89, and it was not significant. However, a subset of patient analysis, they found that patients who had greater than 75% at 2 plus intensity staining in the tumor cells, there could have been some effect. And based on that, a single arm study named Soraya was designed, and with the folate receptor alpha cutoff of 75% at 2 plus intensity. And this study had a response rate of 32%, and it was thought that this will be better than standard of care. And with that, FDA gave the accelerated approval that Dr. Coker was saying. A lot of ADCs are first getting accelerated approval. But then by the time they had already designed, and that's a big phase 3 study was going on, Mirasol, and that also showed, recapitulated all these results. And the final approval was based on that Mirasol study that has a PFS hazard ratio of 0.65. So that was pretty significant when this cutoff was changed. So this is a good example to keep in mind. Now, all targets won't have that. Like, for example, HER2 is a different scenario. Like, it started with high expression, and now more and more, and HER2 is being has been approved for lower and even ultra low. Now is being tested. So again, it depends on the target.
Agreed. And I love that story because I had the great luxury of working on that program from day one. I remember the first in human protocol was just a general all solid tumor all comers to begin with. 8 years later, it's beautiful to see that ADC on the market helping ovarian cancer patients, and it's a pretty significant development in that space. So thanks for sharing, because it's also a good case study and warning of thinking about your biomarker strategy early. I think that's a good topic to maybe shift into, Stephanie, from an operational perspective. Certainly get that question. ADCs are directed at specific antigens. How do you think about biomarker strategy? When do you start selecting patients? How do you build that biomarker selection criteria? What's kind of your best practices in that area?
Yeah, so I would say, it really depends on your target. If you're starting from scratch to develop a new assay, this can actually be a quite lengthy process. In some cases, for example, let's take an IHC assay. It can take anywhere from 6 to 8 months for development and validation. So really, there's a lot of translational and operational considerations when you're implementing a biomarker patient selection strategy. Some of the questions that you'll definitely want to ask are, how will positivity be defined? How will the scoring criteria be? And if you're using percent expression, what will the selection cutoff be? We heard from Dr. Guha that these are not always straightforward, because in some cases, you really need your clinical data to inform you of what these cutoffs or expression levels should be. And if this is a new assay, you may actually just want to consider doing retrospective staining, especially for phase 1 or part 1, because this can help you really better understand, as Dr. Coker was saying, your expression to response relationship in the population that you intend to treat. And, also, if there's an assay that's already in use, an expression level or cutoff might already be known. But regardless, from an operational perspective, one thing that you can consider doing is actually adding a pre-screening portion to your clinical trial. And this will allow you to identify, we'll call them the right expressors. And so you can find the patients that you want to move forward to screening or screen fail. This is particularly helpful when your specific biomarker expression might be lower than others, and so your expected screen failure rate can be high. So this can help weed out some of those lower expressors who may not have the best opportunity at responding to your specific molecule.
Agreed. How about Dende? What's your view on biomarker strategies and how to think about that
Yeah.
early?
Yeah, Stephanie captured it well. I could just build on by saying, um, the target really matters. Again, whether it's a validated target or whether it's a novel target. HER2, I'll use that as a case study where we have decades of experience with that as a target in breast cancer and gastric cancer and other indications. So there could be a commercially available assay that's available, and I think it's reasonable to piggyback on some of those assay rather than starting from scratch. If you're starting from scratch, then it's a whole different ball game. You have to build the science from the get-go. You want to know, the expression levels of the target in your target population. You want to build a robust assay to be able to detect the biomarker, have it validated. And that's a lengthy process, like Stephanie said. So really, in short, validation determines when and how you incorporate these biomarkers, whether it's validated or not. And then that will determine how you want to de-risk your strategy moving forward. I think that's pretty core to the question here. Is the biomarker validated? Is it not validated? And the regulatory path you need to pursue to show that it's relevant or not relevant to the population you're targeting.
Yeah, agreed. I hope you guys don't mind. I'm looking at the clock, and thank you to all our attendees who've been sticking around. Almost everybody's still here from the start. There's a couple topics I want to make sure we hit because from my view, getting the fun job of seeing every ADC protocol that comes across Worldwide's desk, we get these questions every time. The landscape we were talking about, there are thousands in development. There are thousands of active clinical trials. ADCs are very popular. It's becoming a fairly crowded, competitive trial landscape. So how do you navigate that in today's world if there might be multiple very similar ADCs already out there? How global do you need to get out of the gate, or at what point? For my worldwide friends, what's the global tude of your trial look like? Stephanie, kind of curious of your view there, because I know you've been exploring that, on some recent trials. How do you tackle the competitive landscape?
Yeah, thanks, Andrew. So I would say you as the sponsor and drug developer know your molecule best. And so if you have a unique linker technology, a longer half-life, better safety profile, you want to play up what your advantages that the product you're developing can offer that others have not or can't, right? So the clinical sites are getting more and more experience with ADCs. As we heard, they're becoming very popular, and they know how their patients are feeling on them and how they can tolerate their toxicities or how long they can be on treatment for. And so this allows you the advantage to really focus on those areas and help clinicians and patients become more interested in your clinical trial as a treatment alternative. So this, again, goes back to understanding your competitive landscape, doing your homework, keeping up with data disclosures, publications, scientific conferences of those that came before or alongside you. When it comes to going global, I think this is a very unique decision for your organization and based on the circumstances of your trial. But one strategy that I could offer would be really starting just small and local in part one, going through dose escalation, understanding how your molecule is working, what your toxicity looks like, your efficacy, and then leveraging a more global strategy for part two, especially dose optimization, truly once you have proof of concept that has been achieved.
Agreed. I see that as a pretty common theme with most of the protocols that I've gotten to work on. Dende, how about your perspective from the competitive landscape? How to stand out if in a crowded field?
Yep. I think, having a clear understanding on the landscape, knowing what's already approved, what could be coming, will be essential in positioning yourself. Knowing what the gaps are in the current knowledge and the current thinking of already approved drugs or drugs still in development, then you'll be able to position your molecule. Is your molecule able to address some of these gaps? For example, is your ADC able to deliver a better safety profile compared to other drugs. For example, Trodelvy has very high rates of neutropenia and diarrhea that comes in a black box warning. So ADC able to address that safety gap. I think, knowledge of your molecule, how we can differentiate the population, and also the footprint of already approved ADCs or emerging ADCs in development would give you an idea how to strategize and how to place your products while you're developing it.
That's perfect. All right, look at the clock. One more topic that I think is pretty important, and we hear all the time. We've talked about the toxicities and safety profiles of ADCs with some of these payloads. It's fairly unique. There were some toxicities in early ADC development that people did not expect, like ocular toxicity to me was a novel one. Why would a drug specifically affect the eye and make your vision blurry, for example? Let's hit that, and then I want to make sure we have time to get some questions from the audience and go back to Corey to wrap things up. So Stephanie, let's talk about safety and SAEs and toxicity with ADCs real quick. What's your view?
Yeah.
What's unique to this space?
So again, we've talked about this topic over and over, but really understanding what other ADCs with similar payloads have experienced is going to help you figure out how you can manage and monitor those toxicities. But at the same time, it's extremely important to understand, based on your specific molecule's preclinical tox studies, what the data actually shows. So for example, if you didn't see any ocular toxicities like you were mentioning, Andrew, there may be no reason to exclude those patients from your clinical trial or to require prophylactic measures up front. You may want to just get a better understanding for how your unique molecule works in the clinic before you start implementing too many restrictions or requirements, because obviously that can impact your ability to enroll.
And then Stephanie, how do you kind of mitigate some of those things that you might be expecting given your payload or your linker or your target technology?
Yeah, so that's the $1,000,000 question. You really need to train your sites on how to identify these events of clinical interest. And again, if there was preclinical evidence that you are or may observe certain toxicities, then you want to make sure that your protocol accounts for ways to monitor for them. Let's take pulmonary toxicities, for example. You may add pulse ox collections with each vital sign, require a chest or CT or MRI at baseline for all of your patients so that you can monitor any potential occurrence of these events. And then also you could require mandatory pulmonary consults if there's suspicion for things like pneumonitis or ILD. Having guidance in your protocol about how to manage dose reductions or dose delays. One thing is, if you do see certain toxicities that you want to manage upfront, you may implement different prophylactic measures, let's say eye drops for your ocular toxicities or mouthwash. Then you'll want to consider how you're going to source those items. It could be via a centralized vendor. You could have each trial site source on their own. But obviously, there are many operational considerations here. So sites, it's a burden for them to be having to source certain things on their own. But on the flip side, if you're using a centralized vendor, you could run into supply chain issues. And I know we all know what happened during COVID. This is definitely something that where we saw supply chain issues. Another thing is, if you are going global with your sites, making sure that there's standardization across sites and the supplies that they're able to procure. And then, of course, there's a cost and reimbursement factor to consider. Lastly, I'll say, I think one thing that's extremely helpful from a sponsor's perspective is having the infrastructure to look at data in different ways. We're likely most familiar with things like data visualization tools, Spotfire, BI. Those can be tremendously helpful. And I would say, don't just focus on DLTs or ECIs. While those are usually high grade and more severe, there's also things that, I think Dr. Coker mentioned. You may start seeing trends in more mild or moderate AEs that at first seem not important, like, nausea or vomiting or diarrhea. But those can actually be indicators that may need your attention or further review, as they could give you insight to the tolerability of your drug.
Beautiful. Dr. Guha, Dr. Coker, we could talk for hours, I think, on this topic. Looking at the clock, I want to make sure we have some time to get some input from the audience and make sure we're wrapping up, being mindful of everyone joining us today. So, Corey, maybe I can toss it back to you and we can look at whether there's some questions and then make sure everyone's got some final logistics to wrap things up. That's okay.
Absolutely. Thank you so much, Andrew, and thank you to all our panelists for that really great discussion. Yeah, just looking at the clock, I think we'll have time to hit a couple questions from the audience, but rest assured there will be an opportunity for follow-up after the webinar today. So if you did ask a question and we don't have time to get to it, we'll of course pass that on to the Worldwide team.
And you can also reach out to the Worldwide Clinical Trials team by visiting the webpage on the screen for any questions you may have about this topic. So
I'd like to jump to one of the questions we received here. And it's a question that I think we could talk about for a long time, but maybe we'll get the chance to hear from all of our panelists on this one. This audience member was asking about, just looking into that crystal ball you may have, what do you see as the overall future for ADCs? Will the growth continue? Will there be other applications that you foresee, such as, combinations with immunotherapy or multiple targets and payloads? I think it's a great question to hit now, and what you see for the landscape of ADCs in the future.
Can I go first? I love that type of question because we get on the service side, we get all kinds of exciting opportunities coming across our path. Just really quickly, I think we even saw it this past weekend at the World Lung Conference, if you look at that as an example, or some of the newer data. I think because antibody design and engineering is so well known and understood for decades, we're seeing people now getting really creative, right? There are bispecific ADCs, there are bispecific T-cell engaging ADCs. It doesn't have to be necessarily be a chemotherapy payload anymore. It can be an immunotherapy payload. And I think that's the future where we'll see kind of combining some of these moieties into a targeted and immunotherapy attack that can hit potentially multiple targets. And we're already seeing that in the clinic. There's, trispecific, quadrispecific. You can get nuts with your ADC design. The limit is how big your molecule gets at some point. But that's kind of my view, is where is the kind of the confluence of different mechanisms of action all in one, and you can do that with some of the engineering side of things. Udayan, how about your perspective? What are you seeing in the crystal ball?
Yeah, I mean, I would just add that the combinations will be very important for ADCs. Obviously, the ADCs are approved based on monotherapy activity, but we already have approvals based on immuno plus ADC, like especially with the MMAE ADC combination, but more so also like ADC combination with two different targets, different payloads, and all that. These will come up. Already there is a study like just called the DAT study, like a dual ADC conjugate study, right, with Trodelvy and Padcev in urothelial cancer, where essentially it is the combination of response rates of each one that were approved in that indication and about a 70% response. And also increasing the survival. So at the end, it has to be survival increase, and that's where the combinations will come in.
For sure. We got less than a minute. How about Dr. Coker? What's your take?
I think the field will continue to see a lot of growth. I see where payloads that have previously not been able to be conjugated to ADCs will become conjugated as the technology improves. We have lots of cytotoxic agents out there that are pretty, well-known safety efficacy profile that could serve the ADC platform very well once they could be successfully conjugated to ADCs.
Beautiful. Thank you, guys. Appreciate everyone. Corey, you want to wrap us up?
Yeah, excellent. Yeah, thank you so much, all of you, for your insights during that Q&A, very brief Q&A session there, as well as during our presentation. Unfortunately, yeah, I think we've hit our time, so we'll have to wrap up there. But of course, as I mentioned earlier, if our audience members have any further questions that they'd like to ask, please feel free to reach out by visiting the webpage on the screen. and our team here will be happy to discuss with you after we go offline from the webinar today. I do thank everyone for participating today, especially those who voted in our polling questions. If you are attending via GoToWebinar, please make sure to download the supporting documents that you'll find under the handout section. We have really three great documents there from the worldwide team that should help as supplementary material to our presentation here today. I've also added a link in the chat box where you'll be able to view the recording of this event, and you can of course share this link with your colleagues so that they may register to view it as well. You'll also be receiving a follow-up email from Xtalks that gives you access to this recording, and after we wrap things up here, a survey window will appear on your screen. Your participation in the survey is always appreciated as it helps us to improve our webinars. Just a quick note for those of you watching on LinkedIn Live, we do encourage you to create an account on xtalks.com and register for this event, which will not only give you access to today's recording, but to our entire library of content, which includes some previous webinars from our worldwide friends here as well. So definitely register with Xtalks and you'll be able to check that out. Before we sign off, I'd like to thank Andrew, Dr. Guha, Dr. Coker, Stephanie. Really appreciate your time here today. We hope our audience found it insightful and informative, and we wish you all a great rest of your day. Thank you so much, everyone, and take care.
Thank you, everyone.
Thank you.